I don't have a big intro for George. I can say that I took his biophysics course back in about 2001 and was very impressed and remain impressed. So let's give a warm welcome to George.
Yes, it's wonderful to be here. Since this is supposed to be a breakfast dialogue, that means I'm going to try to minimize the monologue part of it, which means I'll go through some of these slides a little bit faster than might be comfortable, but you can always go back and we can drill down on whichever topic captures your imagination. How's that sound? Okay? And I will ask you some questions along the way, like I just did. This is my conflict of interest slide. I'm sorry it's incomplete, but it's the best I could do. It fits on the slide. And you can see Cray and Seagate up there. This is and that's also a thank you slide, because that's the village that it takes to get some of our crazy ideas to a point where they can actually make an impact on society. This is one of those crazy ideas from about a decade ago, the idea that you could share data about human beings. It was even hard to share it back to the individual. If you had something that could impact their health, the standard of the day was that they were officially de-identified and you could not connect them to their records. Anyway, that's changed radically now, I think in part because of this project, which is personalgenomes.org, openhumans.org, and various partnerships we had. Only a few of them are listed here. And anybody in the world is welcome to participate. It's now centered in 5 different countries, including the United States, Canada, England, Korea, and Austria. And that number keeps increasing. It's not intended to scale, although we'd be delighted to have 7.5 billion people involved in it. And some of the things I'll mention, some of the experiments and technologies we use have been tested on that cohort first. And so whenever you see the initials PGP, that's that cohort, Personal Genome Project. So first, I'm going to tell you how inexpensive and many different things we can do with human genomics, reading and writing DNA. But first, I just want to get a baseline whether it's actually useful. And the way I assess that is if it's useful to you, you would have your genome right now. So how many people here in this room have their full genome sequence? This is astronomical. I can tell you that yesterday when I asked this question, there was nobody other than me. So that's a pretty steep curve, or it could be ascertainment bias. But anyway, these are some of the uses for it. If you are carriers, if you and your spouse are carriers of Tay-Sachs or thousands of other genetic diseases, you can get preconception genetic counseling or post-conception, either via in vitro diagnostics or prenatal. And this prenatal non-invasive testing is the fastest growing diagnostic in history by a long shot. Just a few years ago, essentially nobody had tried it, and now other outside of research, and now there's 3 million going on 4 million families that have used this. And it just involves circulating blood of the fetus in the mother's blood. So it's a very, very non-invasive test. And then if they make it all the way through this, then there's newborn screening, and a surprising number of parents are unaware that for decades, in fact, probably maybe even everybody in this room has been involved in genetic screening in every hospital in the United States and most industrialized nations do this. So 4 million kids per year in the United States get some sort of newborn screening of at least 40 genes, and that number is going up and will probably be whole genome soon. If they make it through the newborn screening, then some fraction, maybe about 3%, have some developmental delay or other medical oddity, which is very hard to diagnose because they're individually rare, collectively common. 3% is a pretty high medical impact on healthy children, on otherwise healthy children, and so then they're their parents start what's called a medical odyssey or genetic odyssey, where they go from physician to physician, specialist and so forth, trying to figure out what's wrong with their kid. And it's very heartrending, not just for the kid, but for the parents. They blame themselves that they did something wrong during the pregnancy. And then when they find out that, oh, it's just a genetic, it's genetic and it's not something they did wrong specifically, it's a great relief, even if there's no cure for it. But sometimes there is. So for example, here are the Beery twins who were misdiagnosed with cerebral palsy, and then they went through a whole series of physicians, and eventually one of them, in desperation back then, now it's less desperate, they sequenced their genome and found out that they had a mutation in the SPR gene, which was one of the first cases of that. And the treatment for it are two nutritional supplements that you could buy over the counter without prescription. And this was available long before the Beery twins came along, and it's not exactly clear why it's on the shelves but there are a lot of things on the nutritional supplement shelves that don't make much sense. But anyway, they take serotonin and dopamine precursors, and they're fine now. Before they could barely stand up or focus on anything, and now they're healthy, happy teenagers. Last time I saw them, they were really very with it and gregarious. And then after you get past childhood, then there's a bunch of later onset diseases, including things that eventually are so late onset they affect all of us through aging. And but the classic case here is Angelina Jolie, who had a 85% chance of breast and ovarian cancer prior to a preventative surgery. Now, to fully understand, I think a lot of people felt that, oh, she had some evidence of breast cancer. She had zero evidence that she was a perfectly healthy individual. This was preventative surgery, and it dropped her from 85% to 5% risk. So she didn't have a mammography positive or a lump or anything like that. It was purely based on risk. Okay. So for prevention, you often associate prevention with really simple things that you don't even need FDA approval for, and these often, I would say, are too little. And then you'll be hearing more and more about liquid biopsy, where you have, just like the non-invasive prenatal test, you have cancer DNA floating around in your blood. I think that's maybe too late. I mean, at that point, you're already on a path of a Darwinian battle where cancer typically wins. Now, that's not forever, and I certainly wouldn't say that it's impossible. So I think the future of kind of the big data that we're talking about, the future of medicine, is in prevention. I would hope so anyway. And you want that to be impactful and early. But this is the reality of prevention funding right now, is that this is from the NCI website, National Cancer Institute. They have 1% of their pie for prevention. And this has been true ever since Nixon declared war on cancer, maybe even before that. But I would argue that most of the real breakthroughs, the things that prevent a lot of people from ever getting cancer, are on this list. And I mentioned Angelina Jolie with BRCA1, but there are many women who have this, who say, I don't want my kids to have to go through the preventative surgery. I don't want them to be affected at all. And so they go through this preconception, pre-implantation genetics I mentioned earlier, but specifically for BRCA1 and 2. And then there is this huge success story of epidemiology data analysis where any form of smoke, not just cigarettes, ultraviolet light, other carcinogens, and even infectious agents. A lot of people are unaware just how many infectious agents are known to cause cancer, and there are probably a few unknowns as well. And some of the most famous ones, which we take action on, are things like HPV, human papillomavirus, for which there's a vaccine that's actually given to young girls, with some controversy about that, and boys too. Sorry, yeah. So second, audience participation. How many people here are in favor of genetically modified humans? Depends on your definition. Usually that's the pushback I usually get. So the way I'm going to define it in the next slide is in terms of gene therapy. And the way that most people think of it probably is in terms of germline. And these are different things. So in terms of gene therapy, there are already 2,000 clinical trials that are pretty far along in terms of modifications. These have been, one of them has been approved, Glybera in Europe. There's some question as to whether it will stay approved. There's also one approved in China so Glybera is for pancreatitis and in China for cancer. The bad news is they're $1 million a dose, but but the hope is, and that's partly because they're kind of an orphan drug category, which there was a concern in the pharmaceutical industry of stratification, where you would take a blockbuster drug and say, well, it only applies to 10% of the population. Well, some of these apply to a fraction of a percent of the population, but because the Orphan Drug Act, 30% of pharmaceutical profits now come from these rare diseases, but the consequence is that they cost a lot, and hopefully we can bring that down. And I like to remind people that the alternative to this $1 million a dose, the most expensive drug in history, and orphan drugs are not too different, is this genetic counseling that I talked about earlier, which is $1,000 per person. And so we have a big sort of policy and data issue here, which is how to deliver this, how to get people knowledgeable so they know that this is an alternative, because this you don't need to sell, because as soon as you have one of these severe diseases in your family, you find out how to fix it. But this is harder to sell because it's preventative, and most people will get kind of a blank result, which is good. Okay, these are some of the companies involved in editing, and a little bit more about that in a moment. I mentioned that we might be able to bring the price down, and part of the reason I say that is I've had the personal experience of being involved in bringing down the price of the reading, not the writing, well, reading and writing of DNA. This is a reading curve where we were on Moore's Law, which you're all familiar with, I don't need to tell you about it, and then we weren't. We dropped off of it precipitously, we being genome sequencing, where we went from $3 billion to sort of the 800 for the data, and about $1,000 including genetic counseling. And that should have taken 6 decades if we had stayed on the Moore's Law curve, but instead it took about 6 or 7 years. I'm going to show this in a slightly different form here. I wanted you to see it on the logarithmic plot on the previous slide, but this is a more casual form of it. And the $3 billion was actually for a very poor genome. Great building, but a poor genome, which is meaning it was a half a genome. It was 3 billion base pairs, while each one of us has 6 billion base pairs, half from the mother and half from the father. And if you only have half of that information, it's almost as good as having nothing. Okay, so if anyone offers you half of your genome, just say, no thank you, I don't even want it for free. But a few years later, we brought the price down to around $350,000, and by this time it was a diploid, meaning both, all 6 billion base pairs, well, not quite all, but certainly both copies of the parts you could sequence. It was still missing about 5% of the genome, which people don't know how to sequence or what to do with it. But in any case, most of your genome, 95% of it is available then, and it's available now, but now it's $999. And Veritas is not the only one providing this kind of service, but it's the lowest cost one, and that they stuck their neck out and said, we're providing genetic counseling, meaning that not just raw data or barely interpreted data, but exactly what you would expect to get from medical genetics. And so this involves physicians just like classical medical genetics. The only difference is there is not the presupposition that you have family history, because an awful lot of people are the first in their family to have something, and so requiring that they have a family history is not a good idea. Okay, so now you're going to see a number of companies competing over who can provide this genome sequencing data, which is a true data deluge, because it will not just be your genome, it will be various other things that I'll talk about in a moment. But the 6 billion bases interpreted with vast amounts of public and private information, and so some of them will say, I'm better than company X, brand X, because we have a 50-page report on your genome while they only have a 10-page report. And that may sound like a really great sales pitch at first, but I think the appropriate report length is half a page, right? It's like if somebody tells me, oh, hey, I'm gonna give you directions on how to get back home from the Renaissance. I got a 50-page report on how to do that. I'm gonna say, no, thank you. I got a better way of getting home than that. And so that's, I think there's gonna be an interesting discussion about that. So what's next? When I said there's more than the 6 billion base pairs of your genome, there is, what I think is next is this real-time sequencing. This is going to produce a data deluge from each person. It'll be as if we had the video camera on our cell phone kind of clipped to our chest, you know, like in her, and it's just constantly streaming all this video data but it's also data that we don't normally see. As we walk through the world and somebody sneezes, I think I have a picture of this in just a second here. Somebody sneezes like this, you know, we say, gee, you either say that's rude, or you say, gee, I wonder what that is. Could it be a neural reflex, where nothing infectious is happening, or an allergy? Or could it be, you know, like, Ebola or 1918 flu or something like that, which is truly a pandemic or high threat agent? And the thing is, we really don't know. I mean, how many people here have gone to a physician with cough or sneeze or something and gotten a diagnosis? Isn't that funny? I think it's you know, sad and hilarious at the same time. I've never gotten one. I've gotten some through the Personal Genome Project, but that's not conventional medicine, that's research. And I think it's like being blind. It would be, you know, it literally is, we can't see our environment. We can't see that we're running into something that, allergens or a whole variety of bacteria, fungi, and viruses. But that nanopore data that I kind of whipped through comes from devices like this, which are, by the way, we can go back to that data if anybody's interested, but I just wanted to get through the menu of topics we could have this dialogue about. That is a 70-gram device, and almost certainly will get smaller. I think it will be something that you can clip on that will then have a Bluetooth connection or some other connection to your cell phone, and will stream what's going on. This is they call it real-time analyses. It's not quite as real-time as I would like. I would like something that really takes the air or water or samples from your body and tells you the metabolites, proteins, and nucleic acids in your environment. It's not there yet, but it's getting there. So that will be a source of big data that we'll be seeing more and more of. And these are not just observations. I've kind of tried to stress from the very first slide that this is not just about watching and diagnosing. It's really about treatments. And whole new categories of treatments are coming out through the big data. So the microbiome and metabolome were originally sort of just research observations. They were big data on, as you might imagine, microorganisms, viruses, and metabolites. And they're turning into therapeutics, and I just give you just a tiny subset, highly biased towards companies I'm involved in, full disclosure. But things like when you get Clostridia difficile, and I realize that this is a very subtle medical photograph, but you have some of these companies like Series you provide microorganisms that will compete with this serious one. This is present in many people, but when you take antibiotics, ironically, it causes this thing to flare up and can be painful to deadly. But if you have the right microbiome, which is normally obtained by fecal transplant, which is not well controlled, it's usually from somebody who is willing to give you a fecal transplant. And you can even do it in your home, but it's not the sort of thing the FDA would be excited about, I would think. You want something that's reproducible and you can go quality control on it, and that's what Series does. Another source of big data and large computational tasks that we do has to do with protein design, and I should say protein and nucleic acid design. So here, color-coded nucleic acids are DNA and RNA in red and blue, and the protein is sort of tan. And this particular collection, so my original computational background was in crystallography. So I did this sort of stuff as a teenager. Of course, none of these were known at that time, but the idea is that now we finally, back then it was a dream that 40 years ago, it was a dream that you could do protein design at will, that you could take from first principles a goal and engineer a protein the way you would a car or clothing or what have you. And so this particular set is a very special set because this is kind of a self-referential, kind of a self-compiling compiler, if you will. This is a set, I think the complete set of proteins that we can use for editing genomes. The one that you hear about a lot is CRISPR up in the upper right here, but I think it gets a disproportionate share of the attention. Even I can say that because I was one of the co-inventors of it. But we've worked on many of these things, and I think they each still have very important uses. But the point is, we engineer these based on their three-dimensional structure, and they get better and better. And these are just one particular category. We work with many other categories. And so just to give an example of how we then use the computationally designed proteins that are capable of redesigning the DNA that encodes themselves as well as many other DNAs. Here's an example of what I think will be a growing industry, which I'll call high throughput causality. That is to say, when you get your 6 billion base pairs, there'll be 3 million differences between you and me, and your report will probably come up to 0, that there's no risk factors, even though we know that it's not. But the thing is, we're not that worried about false negatives right now, because if you don't get your report, then you have 100% false negatives. And so if you get anything on your report at all that's actionable, then that's great. But there's a whole bunch of things that are called variants of unknown significance, and this was one of those. This guanine on the X chromosome of this poor child was thought to be the cause of the maladies, the cardiomyopathy, and so on. But this child, like everybody, had 3 million differences from the reference genome, and for that matter, about that number from parents as well. So there's a vast amount of background noise. And so how do you know it's that one? I mean, you might have a few biologically inspired guesses, but the way you prove it is you get rid of that G in cells where the only difference is that G. Now that's a very unnatural situation, is that you have 2 human cells that differ by 1 base pair, and you know where that is. But remember I said PGP is this cohort, this worldwide cohort that we can use for a whole variety of really great data sharing, and in this case, cell line sharing. And so we can make from PGP1, who, full disclosure, I'm PGP1, and so these cells came from my left arm. That took skin cells, turned them into stem cells, pluripotent stem cells, which then can be turned into cardiac muscle. And you'll see in the next slide how beautiful that is. But first we wanted to make 2 versions of my cells, which differ by this one guanine, to test cause and effect is that guanine necessary and sufficient to cause the various disease phenotypes, which include lipid composition, mitochondrial problems, and cardiac issues. Now you might say, well, you know, how is a skin cell or even a stem cell relevant to cardiac function? And the answer is that we can reprogram them now, almost any cell into almost any other tissue or organ. We sometimes modestly call them organoids because they're not exactly the same as organs, but each day, each year, they get a little bit closer to the real thing. So now, Bill Poo was our collaborator who brought us the clinical hypothesis that it was that one guanine out of 6 billion base pairs. And Kit Parker helped us with the organ reprogramming. We normally work on brain organs, sort of brain in a dish sort of thing, and Kit works on cardiac muscles. But together we made this cardiac tissue here, which is this beautiful repeating structure, and it beats about 1 per second. And you can see the impact of one base pair on the morphology, on the contraction, and all the biochemistry is exactly messed up in the way that the child displayed. So this is pretty convincing evidence that one base pair was sufficient. Now you might be saying, well, how do you know you just changed one base pair? You know, maybe some other things got changed and they're really the cause of it. Well, first of all, it would be coincidental if those happened to be that they were random mutations that we caused that happen to have the same phenotype. But the other thing is we now think nothing of, my students think nothing of, if you change one base pair in the lab to sequence the whole genome. And so it's pretty routine that they go around, just to check that one base pair and make sure that none of the other 6 billion have changed, they'll sequence it.
Or can you change it back?
And you can change it back. And so the reverse process of changing it back, and this going forward is a cause and effect analysis, and changing it back is called gene therapy. And so some of those companies I mentioned earlier on, Editas and Intellia and so forth, they basically are doing that now in clinical trials. So yes, you can go back and forth. Now that's an example of a rare inherited disease that was where we wanted to know cause and effect, change a variant unknown significance into known significance. There's a bigger set of diseases where in industrialized countries, wealthy countries like the United States, about 90% of us will die from a disease which does not affect 20-year-olds for the most part. And some people don't recognize aging as a disease, but certainly it's the underlying cause of many. And it is reversible. And so when I use the word aging reversal, some people think I'm talking about science fiction. This is science fact in animals. And I could give you a dozen different experiments, but here's one particular one where you have what's called heterochronic parabiosis, where an old and young mouse are sutured together so they share a circulatory system, and you get multiple organ systems that are positively impacted in the old mouse. These include the skeletal and cardiac muscle, the bones, and the nerves. And not all the factors that are responsible for this are known yet, but there is general consensus that this paradigm, this sharing of blood, is robust and reproducible. But it's only one of, I'd say, about a dozen different ways of getting aging reversal in animals. This has not been tried in humans for reasons that you might imagine. But once we have purified factors, then we'll go into clinical trials just like everything else. Here's a second example and last example where a particular compound which is very core to our mitochondrial function, which is called NAD, or nicotinamide is the precursor, and it goes down about twofold in the aging of a mouse. Now, a mouse dies in 22, or is pretty old at 22 months. And so this is one of many things that goes into decline in the aging of many animals. And so we set out to show that we could change this by gene therapy as well. A different kind of genetic manipulation I was talking about before. We changed one G covalent. We essentially deleted that G from the genome. That was an editing event, genome editing. Here we're going to use the same molecule, the same CRISPR-Cas9 molecule. Oh, by the way, how many people here have heard of CRISPR? Yeah. Okay. Anyway, it'll be like the word plastics in the graduate. Yeah.
I have a question. With the big controversy going on right now over ownership of CRISPR-Cas9, do you have a short view that you could insert?
Yeah, sure. The question is about ownership. So I think that in a previous slide I showed how many different ways there are to do genome editing. CRISPR is just one of them. To me, it's no big deal. I mean, even though I had more of a role in inventing it than any of the other methods. And it's nice, it's getting a lot of attention, but I think it's slightly out of proportion, number one. Number two is with almost all inventions, you need multiple patents in order to practice it. And of the 5 founders of Editas, which was one of the companies I started, and the only one that's done an IPO so far in the CRISPR field, of those 5 founders, 2 of them are involved in this patent dispute. And I should point out, it's not a dispute between those 2 people or even between their universities. It's the patent office just noticed that 2 things came in about the same time and they overlap. And it'll be the last such patent interference in history because the United States no longer uses this. So it's kind of an artifact that I can't believe we're paying so much attention to it, because it's not really a drama of conflict of personalities, really. It's a patent office talking to itself with advice. And it's probably not affecting the industry because the other 3 founders have a whole portfolio of uncontested CRISPR patents. So this is David Liu, Keith Young, and myself. Ours are not under contest. So like to make this thing is more than one key patent. So I don't think there is a key patent. Anyway, that's my personal opinion, and I'm not a lawyer. Part-time, okay. So we now use CRISPR in this case not to edit, but to regulate the levels of one of the key proteins in this pathway. And here, just to cut long story short, you can get up to 47-fold impact on that protein, which results in an 8-fold change in the NAD. And remember from the previous slide, we just needed a factor of 2 to reverse the aging effects. So this is still in progress. This is in the weeds, unpublished scientific research, not a cure for human aging as yet. Okay. Now, you can also, for a third thing that you can do with CRISPR, now going back to the gene editing form of it, is that you can now make, you can target a B-cell malignancy, or in principle a variety of other malignancies, but this is with T cells from another person. And this is one of the first times in history since the invention of transplantation where you don't really have to pay attention to matching donor and recipient. The donors or cells are coming in and killing the B-cell malignancies, but you don't want either graft versus host or host versus graft. And you do that by, and the reason you can get away with it is this is the first time you've really been able to engineer the incoming donor cells in the transplantation. And I don't want to belabor all the jargon here, but you can affect several different genes in the incoming T cells, and only because of that do you have an effective therapy. And this little girl is one of the first patients treated for her B-cell leukemia. Taking that one step further, actually a pretty big step further, if you can get universal transplantation from one person's T cells into another child's body, can you go even further and do it from pigs? Now this is an old a mature field that I didn't start. We were latecomers to this. Started about 20 years ago or so, with the idea is that pigs and humans are well matched for the size and physiology of their organs. Basically any organ you would want from a human organ donor, you could get from a pig, except they're not that compatible in terms of their proteins. They're compatible in terms of their shape, but not the proteins. There's immune response. And 15 years ago, there was about $1 billion investment in this field, and was going pretty well. There was a roadmap to how to deal with the immune rejection, and you can see now that previous slide there, we now have universal donors becoming possible. But what happened is it started to become aware, the community started to become aware, including the FDA, that these pig cells, these pig organs, were producing viruses that would get into the human patient recipient who would typically be immune compromised, and so would become an incubator for this virus, and could either have a bad reaction or could even provide an evolutionary pathway to this virus becoming more pathogenic. And so that was considered an unreasonable risk, and there's more or less been a 15-year moratorium on this $1 billion investment ever since, until CRISPR came along, and we realized that CRISPR was making it easier and easier to do 1 or 2 genes. I mean, it was almost trivial to do 1 or 2 genes, 2 or 3 genes at a time, but 62, which is how many of these endogenous retroviruses we felt we had to knock out, 62 at once seemed kind of daunting. But we did it anyway under the principle that if we didn't try it, we would definitely fail. And that's kind of the motto of our lab is if you don't try it, you're going to fail. And so we tried it, and to our pleasant surprise, just like that sequencing plot that I showed you was like 6 decades ahead of schedule, this, which had held us up for 15 years, took 15 days to knock out all 62 of the endogenous retroviruses. And I want to emphasize that I didn't have like a army of technicians working for 15 days. It was basically a petri place in the incubator doing its thing for 15 days, okay? Which is that the CRISPR was in there one by one, knocking out all 62 of these endogenous retroviruses. Needless to say, there is, I don't have the slide here, but there was big data behind this. Just like my students think nothing of sequencing a genome to get confirm one base pair, they thought nothing of sequencing 4 pig genomes to make sure that they understood what they were getting into, and to get the full collection of endogenous retroviruses and make a phylogenetic tree and decide which was the most conserved place on the virus that they wanted to attack. By the way, I didn't come up with that acronym, PERV. Okay, so here's another big data project. I think this is the last one I'll talk about, but please interrupt. This one was some major newspaper, I forgot to credit it here. I think it was Scientific American, gave this headline, which is a $100 million Apollo project, the brain. This is from IARPA, which is like DARPA, except it's the intelligence community rather than just the defense community. And they wanted, they're a subset of the multiple government agencies, including the NIH and DARPA and NSF and IARPA, that are in this brain initiative that Obama announced a couple of years ago, and that I had some role in initiating. And I was very delighted when the acronym for this, which is the BRAIN, included the word innovative and the word technologies, because I felt that was a good sign that we could really move the dial quite a bit. But anyway, the subset of this, which I think is one of the most innovative one, is the IARPA subset, and they are looking for machine intelligence from cortical networks, which means that they're trying to, this is one of the rare cases where biology might be able to repay its long debt to the computational community, like many of the people in this room. You know, we obviously owe a huge debt to the big iron and the software and so forth. But the idea here is that we are going to analyze a cubic millimeter of visual cortex, 3 different teams, each with about a third of that money, a cubic millimeter of visual cortex in the context of a variety of behavioral tasks, which and integrate all the activity in that cubic millimeter and try to come up with a code by which visual tasks are done. Because we noticed that even though computer algorithms are getting pretty awesome at a variety of human-like tasks, like chess, Jeopardy, and Go, those are kind of human tasks that were pretty mechanical. We just thought they were really cool because humans could do it. And so now machines are getting better at competing with humans who were competing with machines. And then, but things like visual processing, you know, an infant of almost any species can outperform the algorithms today. You know, like finding a face in a crowd of moving people. Anyway, so the idea is to produce algorithms from summarizing the data we get. And so here's jargon here, omics. It's like genomics, proteomics, and so forth. So genomics, transcriptomics, proteomics, metabolomics, microbiomics, and so forth, immunomics. And I'm just going to show you very quickly how we're doing this kind of project with a new technology that produces a ridiculous amount of data. I mean, it's going to make all you guys very, very happy. And this is called fluorescent in situ sequencing. It's something that we started working on in the 90s. I'm embarrassed to say how long it took to arrive, but it arrived in force in 2014. And forget the protocols. It gives us the 3-dimensional information about RNA. So these are 2 cells. Each of these dots is a single molecule of RNA, which you can sequence the same way. I'm sure you all know about next generation sequencing. I've been mentioning it. Normally in next generation sequencing, you would take a bunch of cells or even one cell and splat it open and spread it out through a flow cell randomly. And then you get the sequence information, but you've lost track of where everything comes from. This says, leave it where it is. It's fine where it is. We're going to harvest not just the sequence, but the 3-dimensional coordinates of the DNA, the RNA, and the proteins. And I'll show you each of these in turn, each of these kinds of omics. This is RNA right here, but you can see the nucleus where there's an abundance of these transcripts. This is the instrument. We helped start most of the next-gen sequencing companies, and we collaborated with them to develop this. They each made small contributions. Mostly the device was built with Danaher's cooperation. This is kind of a data slide showing the G's, A's, T's, and C's, how cleanly separated they are, even though this is all done in situ, in cells, in place. These are cancer cells, so we not only provide we sell the instrument and the services, and most of the people using the services so far seem to be interested in cancer, although I frame this in terms of neurobiology. This is a summary of reads from in situ. Remember, this is in cells. These are like a histogram of where you can find the reads, where you find these in the computer that matches, this is a particularly long messenger RNA, 8,000 base pairs long. And you can see there's some places that are more well represented than others. But we kind of freaked out when we saw an entire exon missing, meaning an entire piece of the protein coding region. Fortunately, in this, we went on to the second half of the experiment, and we found that what it was what's called alternative splicing, where different parts of the protein are expressed in different environmental conditions. So that's RNA still. Now let's talk about DNA. This is DNA. This is work from Ting Wu's lab. She's a colleague in our department. And this shows just how much extra data you get in going to super resolution. So this is the data at conventional microscopy resolution with these are the scale bars here. Same image, same position where this glowing thing is overexposed, so you can see the individual single molecules here. that is the same thing. You can see this is just essentially one big blob. Here, you can blow this up further, and you can see down at the sort of tens of nanometer scale. And put that in perspective, these are individual nucleosomes at the sort of 30 nanometer scale. So you can really trace the complete folding of a chromosome the same way that we currently trace the folding of the proteins. And I showed you that slide with all the rotating proteins. That's where we're going with the chromosome.
Professor Church, what is that storm imaging technology?
What about it?
Sorry, is this the same? Is this physics still, or is this the atomic?
This is, so fluorescence in situ sequencing is kind of a bucket term for anything that involves fluorescent microscopy, where you get sequence identifiers that tell you where you are and what you're looking at. So in principle, you can think of all of our pathology and cell biology. In the past, we'd look at something, we might have a couple of colors, like in the past slides would be H&E stains, 2 colors. And in cell biology, it might be 3 fluorescents. But what you really want is to know at every pixel, every voxel, what's there. And so I'm showing you a couple of different methodologies that all have the same property, which is at a particular voxel, what's there. In the previous ones, it was RNA, and these ones, it's DNA, and they're at higher resolution. So the RNA ones were all at conventional confocal resolution. These are at super resolution with storm microscopy.
Dr. Church, the difference between the cryo-electron microscope and this?
Okay, so in that Brain Initiative project that I said was IARPA, I said there were 3 teams. They're all using the same calcium imaging, but they're using 2 different methods for reconstructing the connectome, that is to say the synapse level connection between nerves, where you might have thousands of synapses per neuron. 2 of them are using electron microscopy, and one of them is using fluorescent in situ sequencing right now. Could change. The thing about electron microscopy, your question, is it has one color, essentially various shades of gray. And so when you look at a position in the electron microscope, no matter what kind of electron, you picked a particular category, no matter what kind it is, you can't tell what you're looking at other than by morphology. You can't say, oh, here's something that looks like a synapse. It's hard to guess whether it's an excitatory or inhibitory synapse. It's hard to guess what the neurotransmitter is, and so forth. So if you have a lot of colors, and you can reuse those colors with sequencing, the trick is that you're not limited to the 4 colors that you can spectrally separate. You can reuse those 4 colors over and over. And so you have GATC in the first round. I mean, it doesn't have to be sequenced. It doesn't have to be strictly GATC, but it's a sequence of events. It's a sequence of colors, you could think of it. But typically, it is convenient that it's actually also a sequence of base pairs. But anyway, GATC on the first round, so it's going to be one of those. Let's say it's G. Second round, it's an A, T. And what happens is you get basically 4 to the N colors. So that's the difference with microscopy. I'm not saying anything bad about electron microscopy. I'm just saying that there's something really good about fluorescence microscopy, because it's now getting close to the same resolution. So the cutting edge of this, which I don't think we need to go to for most of these, is about 5 nanometers. Electron microscopy, you can go down to a fraction of a nanometer, but for most purposes, you don't even need 5, you need 50 nanometers, but you need better than conventional. Here's an example of an entire organism that's being imaged, where each of the chromosomes has its own color. You can also assign colors. You can literally assign colors to every few base pairs. And this one was just to show, so you can see each of these cells has its own set of chromosomes right in this nice array, and you can see how the chromosomes are organized. This is relatively low resolution. So that's RNA and DNA. This is proteins, and yet another way of doing microscopy at super resolution. This is called expansion microscopy. And the previous one, the STORM, gets the high resolution by sampling each individual fluorescent molecule multiple times. And so that takes time. You have to do that, but it gives you the centroid, which is more precise than the diffraction limited Gaussian that you typically would have. This is different. Instead of sampling and getting a centroid, you just literally physically expand your sample. And that, again, it sounds a little like science fiction, but what you do is you anchor your sample onto a hydrogel. So here's an anchor point in this little square grid. It's not really square, it's more of random polymer, is polyacrylate, like what you have in disposable diapers. And you know that one of the properties of polyacrylate in disposable diapers is it expands. And this does the same thing. So if you dilute it in water and remove the counter ions, it will expand up to 4-fold in each dimension. And you can do that a second time, and you get another 4.5-fold, and for a total of 8,000-fold volumetric expansion. So it makes much bigger volume. And it's isotropic. So here's comparing two different, one of them expansion microscopy, and the other one of the super resolution methods that just uses the physics of the optics, and you can see pretty nice correlation in the size of various molecules. But yeah, go ahead. No, go ahead. So the question is about how quickly proteins might change in a cellular media. So this has big advantages over what I would call classical proteomics, which might be mass spectrometry. But they both, because one of the things you're doing, first of all, you're collecting in situ, so you get the three-dimensional coordinates, you get the subtleties of who's next to who in the cell, which is lost typically in mass spectrometry. But in terms of the faithfulness of the structure at the time that you sacrificed or surgically cut out the sample, typically it's dumped immediately into either liquid nitrogen or formaldehyde, or liquid nitrogen followed by formaldehyde. And so these proteins are literally fixed in space by covalent attachment. And so all of the biochemistry and most of the chemistry is eliminated instantly. That doesn't mean that there won't be some subtle changes, and many of these we know about. And sometimes there are individual solutions for those, but for the most part, even things like protein phosphorylation state and other post-synthetic modifications are retained. Yeah, and by the way, this is proteins, and the way you do proteins is a little harder than nucleic acids. So the nucleic acids, you literally just have the computer design the probes. Or you can use random probing, and then the computer interprets the sequence that it gets out. Proteins, you don't have either of those tools available. What you have instead are antibodies, and so you're limited by, or you could say there's a revolution coming in these antibodies. They can either be kind of classic antibodies, these Y-shaped molecules, or they can be a smaller piece, just a so-called nanobody, which is about half the size of one of these things. But you could have these antibodies which are then coupled to a nucleic acid, and this is work from Ed Boyden's group, our close collaborator on many projects, including this one, where the protein is coupled to an antibody which is coupled to a nucleic acid, essentially reducing this to a problem that we've already solved, which is the nucleic acid in situ sequencing. So that's how we do proteins. Now, the last of the omics that I'll show you is how we do small molecules. And the way we do that is we turn the small molecule problem into a protein problem, which we've already turned into a nucleic acid problem. So you can see this little simplification that's kind of what programmers and mathematicians do, is they try to reuse their code. And so what we do here is, let's say we want to detect a molecule like progesterone. We use the protein design tools I mentioned earlier that we've used on CRISPR and a whole variety of things. Here's a second example where we use protein design tools to design a new reporter which binds to the progesterone molecule. This is done from scratch. We could go out into the wild and try to find a protein that binds to progesterone and has the properties we want, but instead we designed it. And we specifically make sure it doesn't bind to a variety of related compounds, so that other steroids, digoxin and so forth, that look kind of similar. And so we can go through this, a combination of computer design and laboratory evolution, and we can get this nice clear distinction between progesterone and other molecules, where the property that we get is that it is the protein, the reporter, is degraded in the absence of progesterone, and it is stabilized by progesterone. And so if the reporter is, say, a fluorescent molecule, we can read out the fluorescence to detect this. Or we can detect it with antibodies in the in situ assay that I showed in the previous slide. So we can turn a small molecule progesterone assay into a larger molecule protein assay via antibodies, via nucleic acids. So we come back to this. The IARPA is just one of many applications of this, what I would call omni, meaning all different kinds of omics. The four that I showed you is DNA, RNA, protein, and metabolites. But when you take it to the brain, which is what IARPA is interested in, we have activity, which for this project we're measuring with calcium imaging, behavior, which is just the context, which is typically video data stream. So this calcium imaging data is like a video stream at as high a frequency as you can get calcium data, which is sort of on the order of 10 hertz or so, gigapixels. Video data is the input to the animal, and also what the animal is doing in behavior. Then the connectome is super resolution synapse level analysis. So you might have 86 million neurons in the mouse brain, 86 billion in the human brain. Multiply times 1,000 to get the number of synapses. It's truly a remarkable number, and we're connecting this now back to the activity. We're integrating it, and then we can also collect the cell types, the development, as it goes from an egg to an embryo with a brain. We can trace that lineage. I'm not going to go through that unless you want to. And then we can follow expression, which is what I've been talking about so far, DNA, RNA, and protein in serial sections. Now I'm just going to end and open it up for more discussion on two totally speculative slides that are just meant to indicate where, just for fun. I mean, it's just playful. It could be taken as an insult to computational, current silicon-based and magnetic-based computation. But anyway, here it is that this 20-watt human wetware, who will remain de-identified, in 1905 did a number, 5 papers, any which of which could have gotten him a Nobel Prize. In practice, he only got it for one of them, I think one of the least impressive of the papers. This task is, I think, considerably harder than Jeopardy to reproduce. Second speculative slide. This is where, this is an experiment I did with my own hands. Usually I prefer doing computing, but in this particular case I put on the purple gloves to encode my book, Geneticist, into DNA. Some of you may have heard of this. And the idea was a very specialized form of data storage. This is not Seagate disk drive. This is about the topic of archiving, much of which is done on disk drives. But these are about 10 to the 6 times the density in 3 dimensions. And it has the oldest DNA that's been recovered intact or readable, I should say, is 700,000 years, which is slightly better longevity record than disk drives so far. Anyway, so we have all these various would-be technologies for storing data, some of which are bleeding-edge physics that require liquid helium or other sources of energy. The DNA in principle doesn't require any energy for long-term storage. And we moved way up on this plot where we have information density in bits per cubic millimeter. This has all been framed, of course, to favor the new technology. And then on the x-axis is the number of bits that have been encoded in production. And of course, hard drives are way out in the terms of the number of bits that are stored in the world, getting into the zettabyte, yottabyte range. But this is where we're now moving our ability to encode digital information in DNA with funding from Technicolor ever since this paper came out in 2012. So I'm just going to leave it at that. Oh, there's this PG Ed organization. I encourage you to go to this website, and we have a space genetics website as well. So I'll just leave this open on this page for additional questions or comments or discussion, dialogue. Yeah. Thanks.
Dr. Joseph, I'm not from this industry. I'm not a biologist, so excuse the language. But you talk about the cost of whole genome sequencing and the counselling that's available, together with that genome. But at the moment, really, the state of the art is a small part of the exome and a bit of counselling around just that parts list, the genome.
Right.
You talk about multi-omics or omniomics here. It gives you the system view.
Yeah.
How do you see the evolution of counselling and reporting based on this multi-omics view?
Yeah. Well, so you're starting to see what I would classify as wellness and concierge medicine going up a notch by including some omics. For example, there's one in San Diego called HLI and one in Seattle called Araval, which I'm involved in, where they are trying to integrate microbiomics, metabolomics, and genomics into a series of slightly more mature technologies like whole body imaging and so forth. Now some of these have raised concern in the past just because of over-diagnosing and hypochondria and so forth. But if you treat it as baseline, where you're getting information on a healthy individual and you're not acting on it, and then when something happens, so rather than saying, oh, I had an incidentaloma, I saw a little blurry white patch in my abdomen, so I'm going to go and do exploratory surgery. Instead, you just say, I'm just going to watch that and see, and when I ever do develop a serious medical symptom, I'll go see what did the baseline image look like, or the baseline genome look like. That said, and when you do have a symptom that locates a particular part in your body that you want to sample, you'd like to do that with the best tools that are available. Each of these things, whether the therapies or diagnostics, are going to have to go through the usual process of safety and efficacy testing, and try to minimize false positives relative to current practice. There were two questions. One is having to do with the reference genome being kind of a half and a mixed genome, and the other is each new genome coming in that's of relevance to you, how should that be done from scratch? That's what I got out of the question. So the reference genome is already evolving into not just a haploid half genome that's a mixture, kind of a monster, to a series of diploid genomes, and in fact, part of this is through a new standard setting, which your agency, the FDA, and the National Institute of Standards and Technology, NIST FDA, called the genome in a bottle, where they're taking trios, meaning mother, father, and child, multiple, I think 8 different trios, most of which come from the Personal Genome Project so far, and now using those to say, at this position, this is the kinds of copy numbers that we see, right? In other words, we're not just going to pick one of them, say from your mother. We're going to pick 24 times 2 at that position. And that'll give us a population idea of what can happen there in healthy individuals, although nobody is absolutely truly healthy. But anyway, you start accumulating this information, and that's the reference. And then for each person, if you assemble their genome from short reads based on the reference, even a better reference that involves 24 people, you could make a mistake, because you're kind of assuming that the scaffolding is right, and it could be this person, came from Mars or Neanderthal or something, and the scaffolding is not right. And so I should add to that the reference genome, even with 24 really well-done genomes, no human genome has ever been complete, which is another warning about the scaffold. I think that's going to change in the next year or so. I think the in situ sequencing is the first method where you literally have connection between telomere to telomere all untouched. You haven't broken it up into pieces at all. It's the biggest piece you can possibly get. Some of these chromosomes are 250 million base pairs long, and the pathetic little Illumina reads are still 300 base pairs long. So I think assembly is a problem, and for the time being, we'll do the best we can. We can do de novo analysis now, but I think in a year or two, that's de novo sequencing, we're going to laugh that we ever thought that was challenging. That's my bold prediction. Yeah, David and I are close friends for many decades, but I would say my version of it is a slightly more extreme version of open, and we have 10 years of experience with it. So the first paper we published on Personal Genome Project was in 2005. In this version, we interact with the individual rather than with large data collection groups. We interact with the individual. We enable them to know what's going on with their own data, so that they're not treated like a lab animal who has no rights. And we also enable them to share it, first making sure that they know what they're getting into when they share it. So a lot of the sharing that's going on right now is with patient samples where the patients don't really know what that means. They are unaware how many instances of even private data are breached, that are released into they should know because of WikiLeaks and other things are very prominent. 26 million veteran records, almost every hospital has been broken into at some point or another. And they should know this, but they don't. In fact, the value of medical records now is higher on the black market than credit cards by a factor of 20. Now you might wonder why I know this, but anyway, I know this. And I'm not selling anything on the black market or buying, and the reason is because a credit card you cancel right away, it's ephemeral, while the data that's in your medical records is permanent and for some reason or other used as identifiers. Your social security number, your mother's maiden name, your eye color, height, weight, and so forth. These are permanent, relative to credit card. Anyway, so they should know that things get out and that things get re-identified. If you have a rich data set, which is almost every useful medical data set is now rich, meaning identifiable. So anyway, so those are the two things we make very clear, and most studies have you just sign a 10-page document that says that really is written by a lawyer for lawyers to protect the institution. Some people think it's for the patient. It has nothing to do with the patient rights. It has to do with the protection of the hospitals and the researchers. We have a mechanism by which they have to answer multiple choice questions to show that they actually understand what they're getting into, which I think is something that should be present in not just in research studies, but in a variety of services as well, whether it's direct to consumer or hospital media. So anyway, once you do all that, if you do all that right, and you've properly communicated and educated the people, and I think that can be done for any person, any country, of any education, if you try, if that's your priority. Now you can share it in a variety of ways. And if you try to share it, anything short of that, I think you're inviting disaster of some sort. That's my opinion. But we made the Personal Genome Project as a demonstration that you could do this, and it's been there. We've had no mishaps in 10 years. Well, I love the fact that more and more are accepting the patient as not only an active component, but the active component. I think that's good. I think the insurance thing is a tough thing to do. What really should probably be done is that all insurance is somehow guaranteed before anybody gets your genome, you or the insurance company. Because if you just allow one of them to have it and the other one not, then you have gaming the system. But the problem is when you get your genome is moving earlier and earlier in time. So it's like, now it may be that most people have their genome are our age, but increasingly you're going to be sequencing your genome in utero. And so then, anyway, so you either have to have a single payer system or you have to have something where the information is not available to insurer. because insurance is supposed to collectively deal with risks. And if you start having really good vision into risks, like all the way down to perfect vision, then you no longer have insurance. And so you really have to go back to either collective at the level of industries or countries. Yeah, so the question is about alternative representations and how many different genomes do we represent at each position in a reference genome. And I think when we first did the genome, that was a much tougher question than it is today, because it was hard to, we had less experience with representations in general, and we had less compute power. But I think with the compute power we have now, basically the representation is the sum of all genomes in the world, which ultimately will be 7 billion or so. And you just have tools that will say at this position, this is what you have. Now, that becomes easier as we get closer to, accurate genomes and comprehensive genomes. We have never sequenced the full genome, and so we have a tendency to hang on to a lot of extra baggage data, meaning that you might store, for just 6 billion base pairs, you might store a terabyte of data or so. In principle, people have shown that you can compress it down to 3 megabytes. So from 3 billion, instead of up to trillions, down to millions of bytes. Now, you still, for searching and so forth, you need to expand it with indices and polymorphisms and so forth. But the point is, we're getting to a variety of representations where you can literally ask, what's all the variation that occurred at that site, and which of those was deleterious for various diseases. So you kind of blur the distinction between doing a simple lookup onto a reference with the more complicated lookup onto full phenotype correlations. And I think that's a good blurring, personally.